Hey guys, welcome back to my channel. So I'm really sorry that I was not uploading the videos because I was not well. Literally I was not well. But today I'm going to give you the question bank as per RGUHS, that is 8th semester regulatory science question answers for 10 marks. This is for 10 marks, 8th semester regulatory science. So the first question or the fourth question in the question bank of regulatory science. So explain the stages in the development of new drugs. If you ever asking me about the questions where I have took, so I have took these questions from the question bank. You can check it out. So the second question is even same stages in the development of new drug. The third question, what is innovator and generic products? And fourth question, explain the stages in the development of generic formulations. And the fifth question, same question as question number 1, stages of development in new drug. Even sixth question is also same, seventh question is also same, and the 8th question or the 26th question in the question bank of regulatory science discuss different stages of preclinical studies. And the 9th question or 29th question same as question number 1 or question number 4, which is given in the question bank of regulatory science. So I'll start from the first question, that is stages of development a new drug. So first we'll start the pre-discovery for the stages of the development for a new drug. We have to understand the disease. What are the diseases that the patient is suffering from to develop a new drug? So understand the disease before any potential of a new medicine can be discovered. The scientists work to discover the disease to be treated and determine the causes of the condition. As I have said you, the pre-discovery means we have to first understand the disease for the patient which is suffering from. So in that, the second, target identification. So we have to target in which part, in which organ, in which tissues the target identification can give the drug. So choose a molecule to target with a drug. So once the researcher have enough understanding of underlying a cause of disease for the pharmaceutical research, select a target of a potential of a new medicine. So a target is generally a single molecule such as gene or protein which is involved in a particular disease. As I have said you, the target is very important for understanding the disease. So target validation. What is target validation? Test the target and confirm its role in the disease. So what do you mean this? Target validation means the target and the confirmation for the disease for making and developing a new drugs. So after choosing a potential target, the scientist must show that it is actually involved in the disease and can be acted upon a drug. So the target validation is crucial to help the scientist to avoid the research path that looking like a promising but ultimately leads to dead ends. So the researchers demonstrate the particular target which is relevant to the disease being studied through the complicated experiments in both living cells and in animal modes of diseases. So research scientists can then identify the compounds that have an effect on the target selected. So this is the drug discovery cycle or the development of a new drug. So the compound collections primary exist through high throughput in vitro, then it goes to secondary assays, counter screens, bioavailability, the toxicity of the medicine and metabolism, where the drug will metabolize, etc. And then it goes to lead compounds as SAR in the sense structural activity relationship, then indirectly or directly it goes to the chemical synthesis, then design, structural characterization of protein ligand, then the clinical candidate. So this is the discovery cycle for the drug or a development of a new drug in the market. So in that, the B, first in A we have know that target validation and target identification pre-discovery. So in B, drug discovery, lead identification. What is mean by lead identification? Understanding the diseases where the scientists are ready to start looking for a lead compound or a substance in one that it is believed to a potential to treat the disease and that could become a drug. So the laboratory scientist can compare known substance with new compound to determine their chances of success. So testing is done on each of the molecules to confirm its effect on the drug target. So there are few ways to find the lead compounds are nature, de novo, high throughput screening, biotechnology, molecular manipulation, serendipity, and early safety test. So in that, number one, nature. Initially the scientists used to turn to a nature to find an interesting compounds for fighting a disease. So the plants and the bacteria are found in the soil where good sources to find a neat compound. So the nature still offers many useful substances, but now here are other ways to approach the discovery drug. What do you mean by a de novo? What is a de novo? With advances in chemistry, the scientists also create molecules from stars. Even they can use the computer's modeling to predict what type of a molecule will work on the disease or the target. And number third, that is high throughput screening. So this process is most common in that the lead were usually found, and the advances in the robotics and the computational power allow researchers to test hundreds of thousands of compounds against the target to identify any that might be promising. Based on the results, several leads compounds are usually selected for the further studies. And number four, that is biotechnology. The scientists can also be use genetically engineered living systems to produce disease fighting biological molecules. And where the fifth, molecular manipulation. In this procedure or the process, the chemical analogues of existing drugs are synthesized, where this is more logical approach and might be yield new compounds with certain advantages. And in that, number six, serendipity. What is meant by serendipity? It is an observation by chance. It means happy observation by chance and has led to the introduction of many useful compounds. Whereas in that lead or the drug discovery, number second, in that early safety test, where the lead compounds go through the series of the test or to provide an early assessment for the safety of the lead compound. So the scientists test ADME, that is adsorption, distribution, metabolism and excretion, that is toxicological properties for each lead, where the toxicological studies or ADME studies are performed on the living cells animals via computational models, where the successful drugs must be absorbed into the bloodstream, distributed to the proper site of the action in the body, metabolized efficiently and effectively, successfully excreted from the body, and lastly demonstrated to be not toxic. So these studies help researchers to lead compounds early for the discovery processes and the stages for the development of a new drug. And the third one, that is lead optimization. So alter the structure of the lead candidate to improve the properties. So the lead compounds that survives the initial screening are then optimized or altered to make them more effective and safer. By changing the structure of a compound, the scientist can give it different properties. For example, they can make it easily or lessly likely to interact with each other chemical pathways in the body, thus reducing the potential for the side effects. And even at these early stages, the researcher begin to think about how the drug will be made in the formulation which will be suitable for the route of administration, etc. And next question, what is generic and innovator process with the generic drug and the generic drug product development? So what is the generic drug? It means a drug product that is comparable to a brand or a references listed drug product in the dosage form, strength, quality, performance and characteristics and intended to use. So all approved products, both innovator and generics, are listed in the FDA's food distribution and approved by drug products with therapeutic equivalence evaluation, that is orange book, where the innovator drug is the first drug which is created containing its specific active ingredient to receive the approval for the use. It is usually the product where which efficacy, safety and quality have been fully established. So the generic drug are labeled with the name of manufacturer and adopted name, non-proprietary name of the drug, where the generic drug must contain some active ingredients as the original formulation, and these are produced and distributed without an patent protection, where the generic products applications are termed as abbreviated because they are generally not required to include a preclinical and clinical data to establish the safety and the effectiveness. As per US FDA, the generic drugs are identical or within a acceptable bioequivalence range to be branded product. So the one way scientists demonstrated the bioequivalence is to measure the time it takes to take the generic drug to reach the bloodstream, exactly 20 to 36 healthy volunteers, where the rate of absorption or the bioavailability for these generics compared to the innovator drug, and the use of bioequivalence on the basis of generic version by Waxman Act 1984, and the availability for the generic products increases the market competition and sustainability lowers price for the branded products. And what are the similarities between the innovator and the generic drugs? So the generic drugs must contain same active ingredients as the innovator products, where the generic product must have same dosage strength, example 500 mg, in comparison to the innovator product. Generic product must be same of dosage form. Generic product must be same in the route of administration, and the generic product must deliver the same amount of drug to the bloodstream as that innovator product. Where the dissimilarities between the innovator and the generic products may have different sizes, shapes and colors, where the generic drug might have different excipients, as well as cost of generic product is less than the innovator product. Where the question number 3 or the next question is steps involved in the generic product development. So explain the stages in the development of generic product. In that stage there are 12 stages for the development of generic product formulations. In that, the first stage is the literature search, 1 patent evaluation, online computerized search, references or standard book. Stage 2, active sourcing like active material prepared list for supplier and check DMF, that is drug master file. And the third stage would be purchasing of API in the sense active pharmaceutical ingredients. Third stage is purchasing the active pharmaceutical ingredients. Purchase active materials from suitable vendors. And stage 4, that is active testing, that is evaluation of active pharmaceutical ingredients like various physiological parameters like melting point, boiling point, solubility, UV analysis, etc., to compare the standard of product with supplied COA. And the fifth stage is innovators product purchasing and testing. In that there are three types, that is physical parameter, dissolution profile, and evaluation of innovator formula for the ingredients, and microscopical evaluation and chemical testing. So the stage 6, that is selection of excipients, which excipients might be preferable for the drug. So on the basis of suitable excipients, the check its compatibility with the drug by performing the physical mixture. And the stage 7, analytical method development and validation. So develop the suitable method for quantitative to contain a drug in a sample which is obtained during various tests for the formulation. And stage 8, formulation development, where the pre-compressional evolution and post-compressional evolution dissolution may occur in the formulation for the development of a drug molecule. And stage 9, optimization of process, which is performed by a process of various parameters and determines its effect on the comparison, hardness, friability, dissolution, disintegration, etc. So prepare the process of optimization report. The POR forms part the product development report. In the stage 10, comparative evaluation of generic product, where it performs the bioequivalence study to develop the product and compare the result with the innovator's product. And stage 11, scale up, technology transfer and validation, whereas validation for the report, which plays an important role for the product development report in the generic formulations. And lastly, ANDA pre-submission auditing. What is ANDA pre-submission auditing? Filling the ANDA checkout availability for all the documents which is given below. Audit all the raw data supporting the development report, and audit plant and laboratory documentation as per ANDA SOPs, standard operating procedures and optimization data. And lastly, IVC report, product process validation protocol scale up. And stage 30, that is ANDA submission, where while filing ANDA, generic company has to choose one of the following option: para 1, para 2, para 3, para 4. Where lastly, the stage 14 is acceptance of ANDA and marketing for the generic products. It's where the next question for the 10 marks in unit 1, preclinical stages. So different stages of preclinical stages. So the next question will be discuss the different stages of preclinical studies. So the preclinical studies in the sense lab and the animal testing to determine the safety of the drug for human trials, where the scientists carry out in vitro and in vivo tests. So the in vitro tests are experiments conducted in lab, usually carried out in the test tubes and beakers. Vitro is a glass in Latin, and in vivo studies are those in the living cells culture and animal models. Vivo is life in Latin. In the Latin word, vivo means life, vitro means glass. And the three major areas for preclinical evaluations of stages: acute, subacute, and chronic toxicity; therapeutic index, that is safety and efficacy evaluation; pharmacokinetic studies, that is ADME, absorption, distribution, metabolism, and excretion, where during these stage the researcher also must work out how to make the large enough quantities of the drug for the clinical trials. Techniques for making a drug in the lab on the small scales do not translate easily for the larger production. So this is the first scale up for the drug will need to be scaled up even more if it is approved for use in the general patent population, where the pharmaceutics, the result of preclinical testing, are used by the experts in the pharmaceutical method to determine how to best formulate the drug for its intended clinical use. For example, a drug that is intended to act on the sinuses may be formulated as a type of time capsule release or as a nasal spray. After starting with approximately 5,000 to 10,000 compounds, scientists now have candidate drugs which will be studied in the clinical trial. And the stages for development of a preclinical, in that IND, that is investigational new drug application, and safety, which is come under the preclinical stages of drug development, where before any clinical trial can be begun, the researcher must file an investigational new drug application with the FDA. The application includes the result of the preclinical work, the candidate drug chemical structure, and how it is thought to be work in the body by listening any side effect and manufacturing information. And the IND also provides a detailed clinical trial plan that outlines how, where, and by whom the studies will perform. Even the FDA reviews the application to make sure the people participating in the clinical trials will not be exposed to unreasonable risk. In addition to IND application, all clinical trials must be reviewed and approved by the Institutional Review Board IRB at the institutions where the trials will be take place. And the process for these includes the development of appropriate consent, which will be required of all the clinical trials participants. Statisticians and others are constantly monitoring the data as it becomes available, and the FDA or the sponsor company can stop the trial at any time if the problem arises, where finally the company sponsoring the research must provide comprehensive regular reports to the FDA and IRB for the progress of clinical trials. So here we have completed the 10 marks of regulatory science 8th semester first unit. Tomorrow I am going to give you the second unit, and I'm saying really sorry that I am not uploading the videos because I was not well. So tomorrow I'm going to give you the second unit of pharmaceutical regulatory science. So please do like, share, and subscribe to my channel.